Hello, everyone. Thank you for your interest in our work on deep learning for diagnosing patients with rare genetic diseases. Here is our agenda for today. We'll go through the motivation of the project and the data we're using. I'll briefly explain how our methods work, and then we'll go through 3 key results. Rare diseases affect 1 in 20 people worldwide, and yet each disease affects no more than 50 per 100,000 individuals. Rare disease diagnosis is challenging due to the considerable heterogeneity of clinical presentations and the large number of candidates that must be considered to identify a molecular diagnosis. Many patients suffering from rare diseases are undiagnosed, and it takes 4 to 5 years on average for patients to receive a diagnosis. So our key question for this work was whether we could leverage deep learning methods to accelerate the diagnosis for rare disease patients. We've seen deep learning methods achieve tremendous promise in other diagnostic areas. So it's a natural question to then ask whether we can apply these approaches to rare disease diagnosis. The challenge is that these advances crucially depend on the existence of large annotated datasets containing thousands of examples per condition for training deep learning models, whereas datasets for rare diseases are 3 orders of magnitude smaller. AI diagnosis of rare diseases encounters challenges that are distinct from those other cases that I showed you. Given the lack of large annotated datasets, the approaches must be able to learn from limited data. Approaches must also be able to extrapolate beyond the training data to atypical disease presentations or never before seen genetic conditions. So let me give you a better idea of this challenge using the patients from the Undiagnosed Diseases Network, or the UDN. The UDN is a multiple site program in the US aimed to diagnose patients with rare diseases. Out of the 465 diagnosed patients in the UDN, there are 378 unique causal genes and 299 unique diseases. So most genes, phenotypes, and diseases are only seen in a single patient. Even within the same disease, patients have considerable phenotypic heterogeneity, sharing only 67% of phenotypes on average. And only 28% of patient phenotypes are known to be associated with the patient's causal gene, indicating that these patients have atypical presentations of known diseases or completely novel conditions. To address these challenges, we created Shepherd, a few-shot predictor for hard-to-diagnose rare diseases. Shepherd takes in as input the patient's set of phenotypes, as well as a list of either candidate genes, patients, or diseases, and leverages an external rare disease knowledge graph to perform multifaceted rare disease diagnosis. Shepherd can perform causal gene discovery, retrieve patients like me with the same causal gene or disease, and provide interpretable characterizations of novel disease presentation. And these applications are really important because we want Shepherd to be able to be integrated throughout the diagnostic process. After getting a clinical workup, after getting genetic sequencing, during case reviews by experts, etc. To do so, we designed Shepherd to have these 3 key features. First of all, we trained Shepherd exclusively on simulated but realistic rare disease patients. Training on the simulated patients is crucial because the 465 UDN patients in our dataset are simply not enough to train a deep learning model. Instead, we use the simulated patients for training and then use the UDN cohort to serve as a completely external cohort for evaluating our model. Secondly, we model patients as subgraphs in a larger rare disease knowledge graph. Each patient is a set of phenotypes and becomes a subgraph in a knowledge graph. The candidate genes or diseases are nodes in a knowledge graph. We use a deep learning approach based on graph neural networks to generate representations of the patients that are informed by the existing knowledge in the knowledge graph. This step is important because it gives us an opportunity to infuse external knowledge into our model. Finally, we leverage label-efficient approaches to train Shepherd. Shepherd learns to capture similarity between patients, genes, and diseases by training the model to generate an embedding of each patient, such that the patient's embedding is located close to the patient's causal gene, disease, and other patients with the same gene or disease, and far away from irrelevant genes and diseases. Now on to the data. Shepherd is trained on a simulated patient cohort of around 42,000 patients that represent 2,100 diseases found in OrphaNet, which is a rare disease knowledge base. To encourage Shepherd to generalize to new diseases, we perform disease split training, where we assign patients to our train and validation sets by disease. So how does the simulation process work? Ultimately, we want each patient to have the following information: a disease name, the gene causing the disease, a list of phenotypes, and a list of candidate genes that could be responsible for the patient's disease. To briefly explain our simulation process, we first pick a disease that we're interested in and take its phenotype terms and causal gene. We next perform phenotype dropout to simulate potential incompleteness of data, and phenotype corruption to simulate the use of more general terms rather than a specific phenotype term. We add more phenotypic noise, and we add distractor genes and related phenotypes. There can be many different types of distractor genes. For example, genes with similar tissue expression as the true disease gene, genes causing diseases with the overlapping phenotypes, and genes causing diseases with no overlapping phenotypes. So ultimately, the goal for simulating patients are to give us meaningful distractor genes and force the model to learn to ignore phenotypic noise or corrupted phenotypes. We evaluate Shepherd on 465 UDM patients who have received a molecular diagnosis, and each patient in the cohort has a set of phenotypes and 2 candidate gene lists. The patients also have an identified causal gene and a disease. Patients in the cohort have 23.9 phenotypes on average, and the 2 candidate gene lists contain 13.3 and 244.3 genes on average that the clinicians must sort through to identify the causal gene. And we leverage a knowledge graph to incorporate external knowledge into Shepherd. The knowledge graph contains over 100,000 nodes and 2 million edges. And this knowledge graph contains multiple scales of relationships. Here are just some of the sources of data that are found in the knowledge graph. All right, on to the method. So Shepherd is trained in a 2-step process: embedding biomedical knowledge and embedding rare disease patient information. First, the model is pre-trained to embed the biomedical knowledge in the knowledge graph. Here we use link prediction to learn representations of each node in the knowledge graph that are informed by its neighbors in the graph. After pre-training Shepherd, we then apply the pre-trained model to our rare disease diagnosis tasks. The input here is the patient's set of phenotypes, and depending on the task, a set of either candidate genes, diseases, or patients. We first overlay the patient information on the knowledge graph, constructing patient subgraphs from the patient's phenotypes. The candidate genes or diseases are simply individual nodes in the knowledge graph. We then leverage our pre-trained model to embed each candidate as well as the patient's phenotype subgraph. Shepherd generates a single patient phenotype embedding from all of the phenotypes in a patient by performing an attention-weighted average of the phenotypes. At the end of this step, the model outputs an embedding for the patient that represents the aggregated patient phenotypes, as well as the embeddings for each of the candidates. We train the model using a loss function that encourages patient embeddings to be close to the embeddings of their causal gene or disease or other patients with the same causal gene or disease. These notions of similarity that are captured in embedding space can be used to rank causal genes or similar patients or characterize novel diseases. To reiterate, a core idea of the training process is to learn a similarity metric between the patient's phenotypes and either the candidate gene, disease, or patient that is reflected in a learned embedding space. For each task, we want the model to capture slightly different notions of the similarity. On to the results. Given a patient's set of phenotypes and a list of candidate genes, our question is, which genes are most likely to explain the patient's presenting symptoms? We first evaluate Shepherd's ability to identify the patient's causal gene from their candidate gene lists. Here, we first evaluate Shepherd's ability to identify the patient's correct genes from their candidate gene lists. Shown here is the performance on the expert curated gene list. On the x-axis is average recall at K, so the number of causal genes retrieved in the top K ranked genes on average across all patients in the external UDN cohort. We find that Shepherd ranks the causal gene first in 40% of UDN patients, achieving a recall of 0.69 when K is 3 and 0.85 when K is 5 on average. We compare Shepherd to several representative approaches, and we find that Shepherd performs significantly better than all other baselines. Shepherd outperforms the second best approach in ranking the causal gene first by 5% and outperforms other machine learning approaches by 24% or more. We evaluate Shepherd's performance on UDM patients stratified by clinical site, evaluation year, and primary presenting symptoms, and we find that Shepherd can generalize across all of these settings. So I'm going to show you a case study of Shepard's ability to perform causal gene discovery on a real UDN patient. This patient was admitted in 2016 and diagnosed in 2019 with a POLR3-related leukodystrophy. Leukodystrophies are a group of rare genetic disorders that affect the white matter of the brain. And this patient has an atypical presentation of leukodystrophy. To get a sense of how challenging this patient is diagnosed, we looked at where the patient's information can be found in a rare disease knowledge graph. Here is a subset of the rare disease knowledge graph that contains all the nodes in the shortest path between the patient's phenotypes, diseases, and causal genes. The orange node is the causal gene, and the green node is patient's disease. All of the dark magenta nodes are phenotypes that are found in the patient. The challenge for diagnosing this patient is the patient's phenotypic heterogeneity. While some of the patient's phenotypes in magenta are connected to the causal gene in the knowledge graph, there are many phenotypes that are several hops away. Only 28% of the 46 patient phenotypes are directly connected to POLR3A, and 94% of the 205 phenotypes directly connected to the gene are not associated with the patient. Despite this, UDN is able to rank the correct gene in the top 2 from both gene lists. Now on to patients like me. Given the patient's set of phenotypes, can we identify other patients with similar genetic conditions? We find that Shepard learns meaningful patient representations that capture patient similarity. What you see here is the visualization of all patient embeddings in a 2D UMAP space. Each patient point is colored according to the disease category of the patient's disease. You can see that Shepard learns to embed patients with similar diseases near each other. The circles represent simulated patients, and we overlay our other patient cohorts on top of the UMAP. Triangles correspond to UDN patients, and inverted triangles correspond to another external cohort of patients who made their data available publicly on the MyGene2 rare disease portal. And we find that Shepard learns to embed patients with similar diseases near each other as well. Let's take a look at a case study. Here we are ranking all of the patients in our cohorts, over 40,000 patients in total, to identify patients that are most similar to this query patient. The UMAP cutout here shows the location of the query patient's embedding and all of the embeddings of patients with the same causal gene. The patient's embedding is a green star, and the patients represented by green circles have the same causal gene as the query patient. All of the light gray circles are patients with a different causal gene. As you can see, patients with the same causal gene tend to localize within that 2D UMAP space. And listed here are the top 5 patients that are most similar to our query patient. When we look at these top ranked patients, we find that Shepard ranks patients with the same causal gene highly. Here, the patient with the same syndrome as the query is ranked second. Interestingly, we see that the most similar patients identified by Shepard do not have many phenotypic terms in common with the query patient. The patient with the same causal gene has only 2 phenotypes, or 10% in common with the query patient. And yet the 266th ranked patient has 5 phenotypes in common. And this is a good thing. It indicates that Shepard is able to learn an embedding space that captures semantic phenotypic similarity beyond just HPO term overlap. And last but not least, we're going to talk about Shepard's ability to perform novel disease characterization. So given the patient's set of phenotypes and a set of known diseases, can we characterize the clinical presentation based on our current knowledge of rare diseases? Shepard can also learn to embed patients in the context of existing diseases. Shown here is another UMAP, but here we're comparing patients to disease nodes in a knowledge graph rather than to other patients. So each triangle is a UDN patient, and each circle is a disease found in an OrphanEt dataset. So each triangle is a UDN patient, and each circle is a disease found in OrphanEt. Colors represent disease categories, same as before. You can see that UDN patients tend to embed nearby diseases of the same disease category. Again, gray triangles here correspond to novel disease presentations. Here is our third case study of the day, a patient with a novel disease. This UDN presented with a number of symptoms that are related to difficulty with food absorption and developmental delay. The patient was diagnosed with a novel syndrome characterized by pancreatic insufficiency and malabsorption due to a mutation in GLYR1. We compare the patient's clinical presentation to known rare diseases, and we find that the patient's presentation is most similar to diseases under the categories of rare inborn errors of metabolism, hepatic and endocrine, and GI disease. It is notable that the rare respiratory disease category is the third lowest ranked category. UDN clinicians hypothesized that the patient's GLYR1 variants interrupt a pathway that causes a mislocalization of CFTR, the cystic fibrosis conductance regulator, which, as the name suggests, is associated with cystic fibrosis. While the patient has gastrointestinal and pancreatic symptoms similar to those in cystic fibrosis, the patient does not have any of the pulmonary features classic for that condition. We also find that 4 of the top 5 most similar individual diseases are also inborn errors of metabolism, and the diseases are associated with phenotypes that are similar to those that are seen in the patient. To summarize, Shepherd is able to perform multifaceted diagnosis and can generalize to never-before-seen phenotypes, genes, and diseases. Shepherd overcomes limitations of traditional data-hungry machine learning approaches by infusing external knowledge via geometric deep learning on a knowledge graph, training on a large disease-split cohort of simulated patients, and leveraging label-efficient training to learn a meaningful embedding space. By training on simulated data, we can release the model publicly to the community without privacy concerns. And you can see a demo of Shepherd at the link here. Thank you so much for your attention. Have a great day.